Hi, everyone. I want to start off by thanking IEEE and the Engineering in Medicine and Biology Society for the opportunity to present at this virtual conference. My name is Jermiah Joseph, and I recently just graduated from Western University with an honor specialization in integrated science with computer science. I am returning to Western this fall to start my master's in medical biophysics. I'm currently involved in the computational medicine lab at Lawson Health Research Unit in London, Ontario, and my interests are primarily in the cardiac electrophysiology field. Today, I will be presenting my full research paper submission number 528, where I investigated the proarrhythmic effects of electrolyte imbalance in virtual human atrial as well as ventricular cardiomyocytes. So to begin with a bit of background, our kidneys play a huge role in the filtration of our blood. So when the body's natural filtration system is unable to remove waste from the blood and maintain a chemical equilibrium, this can result in a number of complications, including renal failure. The specific chemicals in our blood, such as potassium, sodium, and calcium, aid in conducting the electrical impulses in one's heart. So electrolyte levels that are too high or too low can hinder the heart's ability to generate the electrical impulses and thus lead to arrhythmic conditions. In addition, renal failure not only results in the accumulation of waste molecules, which can induce uremia, but can also result in the scarring of cardiac tissue, otherwise known as cardiac fibrosis. The problem is only exacerbated with the onset of alternans, which can lead to fibrillation. So the primary course of treatment for these patients is dialysis. And so this therapeutic intervention is done with a system that acts as an external version of the kidney and removes the blood from the body, filters it, and then returns the clean blood back into the body. And now, however effective this may be at normalizing electrolyte levels and removing waste, this therapeutic intervention fails to relieve the adverse effects of fibrosis and uremia that is caused by renal failure. So we sought to investigate these pathological effects on the heart in silico using PM3. PM3 stands for precision medicine using mathematical modeling and is a computational cardiology platform developed by Dr. Sanjay Kharche and his lab. It can be used to investigate underlying mechanisms as well as analyzing data. Encoded within PM3 are a number of cardiomyocyte models, which are ODEs that can be solved to provide powerful tools and valuable insight regarding electrophysiological mechanisms. So the two that were used for this study are the human atrial cardiomyocyte and the human ventricular cardiomyocytes. And the baseline action potentials of each are shown in figure 1. We can go back to this. So a bit of a method overview. So the 5 main cases that we ran simulations on were controlled with increased electrolytes, uremia, fibrosis, and fibrosis and uremia combined. And so using these 5 cases, we ran simulations where we varied the pacing cycle length, or the PCL. And then from the data we obtained, we used the final 10 excitations to calculate the action potential durations, or APD90s, and calcium transients. So looking back at figure 1 of the baseline behaviors, we can see that uremia conditions prolong the APD90s of both atrial and ventricular cells. And so that indicates a slower heart rate. On the other hand, fibrosis decreased the APD90s in both cells, indicating a faster heart rate. And so as we varied the PCLs for each condition, we looked at the APD90s before and after dialysis. The top panels of each graph represent the atrial cells, and the bottom correspond to the ventricle cells, and the color coding is described in the bottom. So at pre-dialysis, we said that in general, in the atrial cell, all atrial disease conditions result in shorter APD90s overall compared to the control. In the ventricle cell, we see the onset of alternans, in all cases at low PCLs. After dialysis, we see that the altered electrolyte levels, which is in the cyan color, as well as the uremic case, which is in the red color, seem to have an onset of alternans at near physiological heart rates. After dialysis in the ventricle, we see that the disease have overall shorter APDs after dialysis compared to before dialysis, but also has suppressed the alternans. Looking at the calcium transient restitutions, all the pre-dialysis cases in the atrial cell had very similar amplitudes, but overall less than the control. Apart from fibrosis in the ventricle cell, All other disease cases, most notably uremia here in red, has resulted in higher PCLs compared to the control. Finally, at post-dialysis conditions, we see that the atrium calcium instability in uremia as well as altered electrolytes has occurred. These are the same two cases where we saw alternans in the APD90 post-dialysis in the atrial cells. In the ventricle cell, all the amplitude had decreased after dialysis, and the instability was suppressed to occur at lower PCLs compared to before dialysis. Now, overall, this work was simulated using only a handful of parameters, and these are all based on experimental data. And it is very much so possible that the pathological conditions affect other processes that we were unable to incorporate due to just the limited capabilities of our current cardiomyocyte models. We believe that further development and investigation is warranted. As these simulations were only run on the cell level, other studies will focus on extending it to multi-scale tissues and organ simulations. My colleague's current work on this was also accepted to this conference as paper number 530. The work of my project will also feed into future whole heart studies, where the current work is presented at a poster at this conference as well, number 2037. And the investigation of the predictive capabilities of cardiomyocytes is also being presented as a poster number 2038. So once again, I'd like to thank IEEE and EMBS for this opportunity to present my full research paper. I thank Compute Canada for providing the supercomputers, which greatly aided in the efficiency of my simulation runs. I thank the Natural Sciences and Engineering Research Council of Canada, or NSERC, for the Canadian Graduate Scholarship to fund my master's research, and Canary for its ongoing support of my lab. I'd like to extend a special thanks to LHSC and Western University Services, as well as those whose help has made a significant impact on this project, including Dr. Dan Goldman, Dr. Jeff Frisby, and Dr. Chris McIntyre. Finally, I'd like to thank my lab and my supervisor, Dr. Sanjay Kharche, who is currently driving the in silico research here at Western, for all his guidance and on this project. I thank you all for listening. Thank you.